Very good evening, all the audience of Pharmalok. On the behalf of Pharmalok, I welcome you all. Uh, During this Pharmacy Week Samaro 2021, Pharmalok presents a 10 lecture series followed by a Rubaru Pharma Rubaru program. Uh, this is the last lecture of this series, that is 10th one. Today we have uh, privilege with the presence of Dr. Saurav Sharma sir, and he enlightening us with the topic advancement in rational drug design strategies. Before starting with the Dr. Saurav sir, I just introduce our audience with his profile. Dr. Saurav Sharma sir is working as a head of school at Pharmaceutical Science City University, Ludhiana, Punjab. He has his credential more than 15 years of rich teaching both undergraduate and postgraduate student and research experience in basic and clinical pharmacology. Dr. Saurav Sharma is a dedicated, resourceful, and innovative instructor who strive to help a student change marginal grade into excellent grades and support all the colleagues. He has an excellent track record as pharmacy educationist and research scientist. He has two patents and authored 60 uh, research international research paper and two books with particular emphasis on investigation of molecular intervention on vascular dysfunction in pathology of cardiac, brain, and pulmonary circulation. He has previously served in ISF College of Pharmacy during 2006 to 2017 as head of department, PharmD and Pharmacology. He has guided more than 54 PG students and three PhD students in various research projects. He is an he is the recipient of OD Gulati Award in the area of cardiovascular research in 2009 and 2010, Pharma Recognition Award in the 2020, and Young Pharma Personality Award in 2021. His Scopus profile is also included in AD World Scientist ranking during 2021. He has received major grants from Punjab State Council of Science and Technology (AICTE) Research Promotion Scheme and Indian Council of Medical Research in project of for the project of various biology. So welcome, Dr. Saurabh, and please, uh, I welcome on the behalf of all the team of Pharmalog and our audience in this lecture series. So please, you
Thank you,
may
thank you, thank you, Santosh
you
sir.
may start.
Thank you for a warm welcome, and thank you especially to the team Pharmalok and especially Mr. Amit Jha sir, who has organized such a tremendous event, which is accumulating a very intellectual lecture series. And I have gone through most of the lectures, and they were very knowledgeable for the young generation students. And moreover, the Mr. Jha is always contributing in past also by having pharma summit, and accumulating group of faculty members and all the pharmaceutical teachers and academicians and industry persons. So it's a great effort. So I must congratulate Mr. Jha for such tireless and selfless efforts. Moreover, I also congratulate all the pharmacy professionals on this National Pharmacy Week, which is ongoing. And last month itself was a World Pharmacy Week also. So obviously, pharmacy is, the word is round obviously, and the same is truth for the pharmacy professionals. We meet, you see, among ourselves in this way or that way, and obviously, it is a very jubilant and ever dynamic field which is contributing a lot and which has a lot of perspectives. So, with these words, I think we can start with the uh, presentation. Shall we, Santosh sir?
Sure, sir.
Yeah,
Please
yeah. So, to,
go ahead.
yeah, so the topic of you see my presentation, what I have selected is advancement is in the rational drug design and strategies involved in that. So, this topic I have selected because it's a, you see, era which is having a,
I think there is some technical issue. I request to our audience to keep and calm. Our eminent speaker join with us after resolving the technical issue. Yes, our speaker is coming back. So welcome again, sir.
Doctor, I think it's fine now.
Yes, sir. You are online. Please share your slide again.
Okay, okay. Now it's okay.
It's okay. Just put it in full screen mode. During online session, this type of technical issue coming. Uh, please, in full screen mode, sir, please.
I think echo is coming. Is it fine?
Echo is from your side, I think so.
Mm. Hello.
Echo is from your side, sir. Just put it your slide in full screen mode.
Yes. Again, echo is there. Na? I will again rejoin. Okay.
Please.
I think I can leave the studio. Amitji, I'm leaving and again joining.
Definitely, sir. For the clarity of the telecast. Dear audience, please again Have your patience.
Hello. Now it's okay.
Yes, sir. This is okay.
Yeah, yeah. Fine, fine. ठीक है डॉक्टर साहब ओके
Just
so
sharing
the
the.
top uh, screen is okay shared
No, no, no.
sir
Just अभी अभी नहीं आया है. जस्ट आप शेयर करें.
सर आप अपना स्क्रीन शेयर करें.
अमित जी सर का स्लाइड शेयर कर लें.
हाँ, आपका स्क्रीन शेयर करें. फाइन सर.
done
हाँ
sir done
जस्ट.
thank you
हाँ ओके ओके. दैट्स गुड. प्लीज सर गो अहेड.
okay fine sir so the today's topic I have selected is advancements in rational drug design and strategies involved in this. So we will discuss about the basic traditional drug design and what is actually the rational drug design, then the concepts of rational drug design, then types of rational drug design, then structure-based drug design, and ligand-based drug design and pharmacophore-based drug design. So we will go to step by step and highlighting the features of the traditionally ongoing process and what advancements are there. You see, drug design is the inventive process of finding new medications based on knowledge of the biological target. So it's very important because we are at, uh, functioning in the area of therapeutics. We are working in the area of therapeutics where each and every disorder or disease what we are targeting, we should, you see, investigate its major biological targets. And in those biological targets, we are focusing our medications. So that is a very clear picture, which is objective of a, you see, pharmacy professional to develop a medication, to develop a drug which is acting on a biological target and which will obviously subject to the rationalized use and rationalized designing should be there. So that is the major perspective. So in the most basic sense, drug design involves design of small molecules that are complementary in shape and charged to the biomolecular target to which they interact and therefore will bind to it. So if more, you see, rationalized binding is there, more rationalized targeting is there, obviously the accuracy of the and the preciseness of the action will be more and the therapeutic effect will increase. So the drug design frequently but not necessarily relies on computer modeling techniques, and this type of modeling, but that is the computer based modeling, is often referred to as the computer aided drug design. So the modeling techniques for prediction of binding affinity are reasonably successful. However, there are many other properties such as bioavailability, then metabolic half-life, then lack of side effects. So they are not encountered when you are just having a design of a drug with a target. I mean to say ligand with the target. So these properties, bioavailability, metabolic half-life, or other pharmacokinetic profiles are not taken into consideration, but they are very important, obviously. So the ligand and target are very important. With that, the metabolic effects are also very important. The pharmacokinetic profile is very important, and which is to be taken in care for a safe and efficacious drug. So these other characteristics are often difficult to optimize using rational drug design techniques. Now, I will discuss about some of the traditional drug design techniques. Now, traditional drug discovery involves the origin of drug discovery and that evolved in natural source accidental events. So it was not target based and not much systemized as today. Improved and advancements in pharmaceutical science and technology make it evolutionized to much more systemized modern drug discovery. So traditional methods of drug discovery, which rely on trial and error testing of chemical substances on cultured cells or animals and matching the apparent effects to treatments. And these are some of the, you see, traditional drug design methods like random screening, trial and error method, ethnopharmacology method, serendipity method, then classical pharmacology, chemical structure-based drug discovery. Now, if I talk about the random screening, so it includes random screening of synthetic compounds or chemicals or natural products by bioassay procedures involve two approaches: screening for selected class of compounds like alkaloids, flavonoids, or screening of randomly selected plants for selected bioassays. So the random screening is obviously you can select a whole, you see, bunch of compounds and which are alkaloids, and then you are randomly giving, and random from the random screening, obviously it is a tedious process. And keeping this in mind, you see there is a dedicated random screening also, which was taken in past in history, which should not be forgotten and should be taken into consideration to make a viewpoint of the random screening, the importance of the random screening. Now, dedicated random screening is like natural product screening, except that known single compounds are tested at random in your biological assay screen. So it was in the fashion that Gerhard Domag, who was in 1931, obviously in German. So he screened azo-dyes from the paint factory in which he worked. So in the search for compounds with biological activity, this led to the discovery of the first truly effective sulfonamide. So antibacterial prontosil, a red dye in 1935. Now you see was the contribution of the random screening, but it was a dedicated random screening, and a very important compound based on this. A lot of antibacterial sulfonamides were developed later on. So contribution of random screening, again, one of the very important random screening in the past is from 1960 to 1980. So National Cancer Institute and or of National Institute of Health USA. So they have studied around 35,000 plants species. And obviously 20 years it was invested over there, 1960 to 1980. And it resulted in proving two success stories, which were those of paclitaxel and camptothecin. So this is, you see, another contribution of the random screening, which was done in past and which has given these two important compounds. But obviously, you see how much tedious it was. Around 35,000 plant species were taken, and the time taken was 20 years. Then another point of view is the trial and error method. Now, trial and error method includes berries, roots, leaves, and barks could be used for medicinal purposes to alleviate symptoms of illness. Now, example like in history again, willow bark contains salicin, fever reducing in journal. Then cinchona bark contains quinine, and which was you see well reported for malaria. Then Chinese herbal medicines, which is used to treat many illness like malarial. We have artemisinin, artesunate. So similarly, the trial and error methods were like this. They are taken from the plants and they were used as such. If they are working, so they obviously they are used in the therapeutics. So those were the iron trial, but they also has given in past a lot of medicines which are used till the date also. And digitalis is not far away. So digitalis is also the example of that, which was at one time it was a very important drug for a congestive heart failure. And now also for it is in some of the critical cases it is used in the clinical point of view. Then ethnopharmacology approach now depends on empirical experiences related to the use of botanical drugs for the discovery of biological active new chemical entity. So this process involves the observation, description, and experimental investigation of indigenous drugs. And like morphine from Papaver somniferum. Now, morphine opium derivatives, you know about the opium derivatives, and they are used for analgesic effect. They are used for anesthetic effect also. So they are playing moreover, they are also used for cough and maybe for intensive conditions where GIT diarrhea is there. So obviously the morphine opium derivatives are also used in a lot nowadays. So they were all coming from the ethnopharmacology, from the basics of the, you see, ethnomedicines. They are coming and they are used till that point. So berberin from Berberis, Arstata, and picroside from Picroiza curare. So these are some of the examples from the ethnopharmacology. Now, contribution of ethnopharmacology, obviously the artemisinin from Artemisia alba. So these are some of the examples. Now, Google sterones, then bacrosides from Bacopa minera. So these are some of the neotropic compounds. So they all were, you see, used in the traditional system of medicines, like in Ayurveda or in other Chinese system of medicine. They were used, and they were further investigated for their pharmacological effects, and they are in the current system of medicines also. Then another method is the serendipity method. Now, serendipity refers to an accidental discovery. Now, this is another point of view which give you a very important antibiotic, first antibiotic, that is the serendipitous discovery of penicillin in 1928 by Alexander Fleming. So, now serendipity was very important point where in the history, and which has given a lot of compounds. But obviously you now, if you can see today, you are not, you see, you cannot wait for serendipity, you cannot wait for serendipity for the treatment of COVID. So you have to, you see, go forward, and you have to develop or discover new medicines, and so that the disease, as the diseases are increases, as the disorders are increasing, as the pandemics are coming, so folks should be very fast. So but in the history, such serendipitous approaches have given a lot of compounds which are utilized. So then the classical pharmacology, now also known as function based approach. Now this is another point of view where the experimental induced seizures or lowering of blood glucose sugar level, or suppression of anti-inflammatory responses were tested in the animals, and based on those approaches, the drug target is then you see visualized, and then it is taken into the next table. So it is also based off the classical pharmacology and basic pharmacology, and from that point of view, we are proceeding towards taking it to the next stage of the preclinical trials, proper preclinical trials. So, and obviously they are also some of the compounds which are very importantly, like ergot derivatives, then digitalis is there, foxglove. So some of the compounds were intensively used in the basic pharmacology, and then they are taken to the next table. So here is some of the list of those compounds which were investigated, and then they are taken to the next table. Now another approach was the chemical structure based discovery. Now SAR studies, now our system of you see basics chemistry, it has this important point, very very important. And from one uh, class of the drugs, a prototype of the drug, of further classes of the drugs are forming, and that is the base of this particular structure based drug discovery. So, and in 1891, you see, Paul Ehrlich coined the term chemotherapy, used synthetic chemicals to try and cure diseases. So this is, you see, further going towards the SAR studies, and the SAR studies are giving further compounds. Now, these were the some of the concepts what we are talking, were the concepts of the uh, traditional drug designs, and these traditional drug design methods were carried out. But obviously, these traditional drug designing methods are very importantly giving you the basic concepts, but howsoever
I think there is again some technical issue.
are specific to the target, specific to the disease, and specific to the therapeutic effect. So now this is the concept of rational drug design. Now, rational drug design refers to the development of medications based on the study of the structure and functions. Very important. The point is structure and function of the target molecule. And it basically involves three general steps. Identify a receptor. Now here you understand the difference. Now identify a receptor or enzyme that is relevant to a disease that they are going to design a drug for, the first point. The second point is elucidate the structure and function of this receptor or enzyme. So you should know the structure and function of the receptor or enzyme or any other target, biological target, so which you are focusing. So use the information from step 2 in order to design a drug molecule that interacts with the receptor or enzyme in a therapeutically beneficial way. So these are the, you see, three basic steps which I will emphasize. And here, what is you see focus? The focus is first the target and then the ligand. Simple too. And there are various strategies which are rotating around it. So that is the point we are talking about, the rational drug design. So which ligands to be given, which specific ligands to be designed. And now here the basic requirement obviously is a particularly metabolic or signaling pathway. So that is specific to a disease conditions or pathology, or to infectivity or survival of a microbial pathogen. So now the point of view, what is focus in this particular slide is there are some approaches which are focusing the target, but howsoever the target also depends many times on the pathology, on the overall pathology and the pathogens, maybe in the microbial diseases. So we have to make it in consideration which sort of the target is there, and what sort of the pathology is there, and what sort of the you see pathogens or other risk factors are there, or the causative agents are there which is causing that disease. So that is also very important. And keeping them in mind, the targeting is you see identified. And once the targeting is identified, then we are going for the ligand design. Now, this is the drug design process, which is you see mentioned in this particular slide. And if you can understand it, this first is the you see disease. Again, the disease is very important. Then the target identification, then protein sequencing, then structure determination. Now here, what is focused? Now target is focused. The target's protein structure determination, then 3D structure, then active leads if there are any, then lead search, then lead structures, and led to drug development, then drug candidate, then clinical trials, then finally the drug is coming. So obviously this is a you see holistic approach which is focused in the modern system of medicine and in the you see current rational drug design processes. And a lot of investments are there, lot of you see high tech uh, equipments are being you see in the market, and which are used in the industry. And obviously they have made this particular uh, uh, design process very simplified, very efficient, very you see well focused, and giving you see reliable products also. Now, the type of drug design, obviously, the type of drug design as per the you see advancements, if we talk about it, is structure-based drug design. Again, the structure-based drug design, which are the base of the we have discussed in the traditional also. So it is you see more focused, more relied, more investigating the particular structure of the proteins, and then they are targeted. And similar is the ligand-based drug design. So the ligand and drug you see structure, target structure are both targeted in this particular case. Now, this is, you see, again, the structure-based drug design and the ligand-based drug design. If you can see my slide. Now, here, two types of the virtual screening and de novo drug design is there, where the fragment location method, site point connection method, fragment connection method, sequential build-up method, whole molecule method, random connection method. And in the ligand-based drug design, we had QSAR, we had quantitative structure activity relationship, which is 3D or 2D, then scaffolding hooping, then pharmacophore modeling, then pseudoreceptor modeling. So this is you see a brief picture of the rational drug design process. And if I talk more about this, the structure-based drug design is obviously the techniques, as we just discussed, the techniques like X-ray, crystallography, or NMR spectroscopy. So based on certain techniques, the 3D structures are being elaborated, and those 3D structures elaborated, then the target and the ligand binding is, you see, investigated, and which is giving you see more emphasis on the binding with the you see target ligand target, and what is the change, what is the you see expected change in that, expected outcome in that. So that is more focused. Then you see here structure based drug design, again this is a picture which is showing you the high throughput screening of chemical library, then virtual screening, then lead discovery, then evaluation of biological potency and other properties. So this is another brief picture which is, you see, focusing on the structure based drug design. And obviously the structure of the target is very important. And now the protein structure determination. Now here obviously the target structure is very important, and again the targeting, the study of the target structure is done, and based on that the ligands are, you see, synthesized. And what we call it as a dry chemistry lab. It means like bioinformatics softwares are used, and on those bioinformatics software the 3D or 2D structures are studied, and the interaction of the drug and target is investigated, and the best compounds are screened out from a, you see, bunch of compounds. The best compounds are screened out, and after those screening, then they are taken into the next stage. Then another strategy is homology modeling. Now, homology modeling is comparative modeling of protein, refers to constructing an automatic resolution model of the target protein from its amino acid sequence and an experimental three-dimensional structure of the related homologous protein. So this is the another strategy which is followed in this particular sequence of the structure modeling, where the amino acid structure is being studied, and comparatively the proteins which are similar in structure are taken into consideration as a targets. Then another strategy is high throughput screening. Now, high throughput screening is the pharmaceutical industry has adopted the experimental screening of large libraries of chemicals against the therapeutically relevant target as means to identify new lead compounds. So though HTS active compounds, antibodies or genes which modulate a particular molecular pathway may be identified. Then virtual screening is also one of the strategy which is in the current rational drug design, and in this that derived from experimental data, homology modeling, or from molecular dynamic simulations, the, you see, screening is done. Now these are the simulated programs. Obviously these are the simulated programs which are subjected, where the druggability of the receptor, first of all the target should be druggable, and then the druggable receptor is, you see, virtual screened for the binding of the ligand, and what is the, you see, selectivity, which ligand is better binding, and based on that the screening is done. Then another strategy is, you see, docking. Now docking is the obviously sequential again strategy which is used, where the ability to position ligand to the active or a designed site of a protein and calculate the specific binding affinities. Now here in the traditional, you see, classical pharmacology, the potency and the, you see, base of the efficacy depends upon the binding tendency also. And agonist and antagonist both can bind, but agonist has efficacy and affinity both, and antagonist is only having the affinity, but it does not have the efficacy. So obviously the affinity is very important, the binding tendency is very important. So this docking, make it a point here, the only point of view is the binding capacity. You are not, you see, able to see the efficacy, but just the binding, just the affinity. So the affinity is observed by this docking, and the affinity means binding, the binding site. Now this is one of the picture which is showing you the, you see, binding of a particular molecule to the receptor on the cavities of the receptor. It means the binding site, and the, you can visualize the binding conformations, and based on those binding conformations of different compounds, the best compounds can be taken out. Now, the another point is the de novo evaluation. Now, the de novo evaluation is another, you see, point of view in the rational drug design, in which the ligands can be modified, and the ligands can be modified by the, you see, scaffolds modification, and their derivatives can be made which are complementary to the receptors. So the based on the docking studies, the ligands can be modified, and they can be, structurally derivatized, and then they are subjected to the binding with the particular uh, receptor. So if one ligand is not binding, okay, you are making derivatives, and based on those derivatives, then you are pretending it to be bind to the another structure. So based on that particular derivatization, so there may be the better compounds which can come into the picture. Now this is the same point of view what we have just discussed. So first is the docking scaffolds, then add reactants, substituents, and then find the conformation, the best conformation, and which is minimum score rise, and this is best binding compound. So this is the point what we were talking about, and here the best compounds can be taken out after the derivatization. Then ligand-based drug design. Now, the ligand-based drug design relies on knowledge of other molecules that bind to the biological target of interest. So this is another picture which is giving you very specific terms. The terms are QSAR, scaffold hopping, then pharmacophore approach, and pseudoreceptor. So we will talk a brief about it, because this, point of views of rational drug design is a very broad point of view. So we will discuss a brief about it. Now, the quantitative structure activity relationship, obviously, it is a wider use technique nowadays, and it employs statistics and analytical tools to investigate the relationship between the structure of ligands and their corresponding effects. So, obviously, quantitative, here the term is the quantitative. So, the quantitative, if we are interacting ligand and target, so the quantitative analysis should be there. So if the better compounds are to be screened, so the quantitative parameters should be there, and those parameters are involved in this particular QSAR studies. Now, based on this, we have 2D QSAR, we have 3D QSAR, and further we have some comparative molecular field analysis and comparative molecular similarity indices analysis. Now, if I emphasize in the simple words, comparatively molecular field analysis, it is established on the concept that the biological activity of a molecule is dependent on of the surrounding molecular fields, such as steric or electrostatic fields. So this is, very simple statement which is written here, and it depends upon surrounding molecular fields such as steric and electrostatic fields. So the binding depends upon them. Now, another picture is COMSIA, now comparative molecular similarity index analysis. So it is a method developed recently as an extension of COMFA. Now, the COMSIA method includes more additional field properties. They are steric, electrostatic, hydrophobic, hydrogen bond donor, and hydrogen bond acceptor. So it is a, advanced, which is involving other fields also, in addition to the steric and electrostatic field. So obviously, if one target ligand is to be bind with the target, so the surrounding fields are very important, and there may be the hindrance due to those fields. So those fields which are surrounding it are very important, and which are considered by these particular strategies. Another is the pseudoreceptor. Now, pseudoreceptor models combine the advantages of these two strategies are and represent a unifying concept for both receptor mapping and ligand matching. So they can provide an entry point for structure-based modeling in drug discovery projects that lack a high resolution structure of the target. So I again emphasize the pseudo receptors, which are very important. Pseudo means false. In the simple term, it means false. Now, these models combine the advantage of these two strategies and represent a unifying concept for both receptor mapping and ligand matching. So it is not exactly the receptors which are target, but it is used as models, and those models, based on those models, the ligand matching can be done, and then they can be taken to the next exact targets. So the pseudo receptors are playing a very important role in the studies. Then pharmacophore based approach. Now, pharmacophore based approach is the extension of these approaches, and the pharmacophore is ensemble of steric electronic features that is necessary to ensure the optimal supramolecular interactions with the specific biological target and to trigger its biological response. Now, in the simple words, I will again repeat it. A pharmacophore is the ensemble of steric and electronic features. Steric and electronic features that is necessary to ensure the optimal supramolecular interactions with a specific biological target and to trigger its biological response. Now, it is, you see, interactive. Uh, a very important term is the interaction here. So, the interaction between the biological target, which is very specific, maybe stimulating, maybe blocking. So but the binding is very important. The interaction is very important. So the pharmacophore should be considered as the largest common denominator of the molecular interaction features shared by a set of the active molecule. So thus a pharmacophore does not represent a real molecule or a set of chemical groups, but is an abstract concept. A pharmacophore is the pattern of features of a molecule that is responsible for a biological effect. Maybe inhibiting, maybe activating, but the pharmacophore gives you a picture of the, features, a pattern of the features of a molecule, which is usually responsible for the biological effect. And based on those pharmacophores, you can identify the ligands for the better targeting. And now this is the, you see, some of the pharmacophore fingerprints which are there, and based on these pharmacophore fingerprints, the binding strategies can be designated, and it can be, you see, distinguished. And based on these pharmacophore fingerprints, you can identify the ligands which are of the similar fields, similar interactive fields. So the interactions are investigated based on these pharmacophores. Now, a pharmacophore model consists of a few features organized in a specific 3D manner, and each feature is typically represented as a sphere with the radius determining the tolerance of the deviation from the exact position. And here you can see the hydrogen bond acceptor, hydrogen bond donor, anionic, cationic, hydrophobic, and aromatic part. So obviously in this is very specific, and based on that we can design the ligand, and we can identify the ligand. Okay, so this ligand can be fitted in this, and then we can take it to the next table. Now, the main applications of the pharmacophore modeling is in de novo design of the ligands. It also has its role in virtual screening and docking, compared with pharmacophore based virtual screening, and pharmacophore based de novo design shows a unique advantage in building completely novel hit compounds. So the applications of pharmacophore have also been extended to lead optimization, multi-target drug design, and activity profiling and target identification. So I think these are some of the approaches that we have focused. These approaches are, you see, focusing ultimately the advancements, and it gives you a comparative feature between the traditional and the recent parameters which are considered in the drug designing process. So, Santosh sir, I think it's okay, fine.
Sir, very informative session it was.
Yes, sir.
Very informative session, and it sum up in the small duration. You just Gagar में सागर. I
जी
say this thing. आपने आपने एडवांसमेंट जो रिसेंट एडवांसमेंट रेशनल ड्रग डिजाइन स्ट्रेटजी है, जो ज़्यादातर हमारे स्कॉलर्स इसको अडॉप्ट करते हैं, उसको आपने काफ़ी सिंपल वे में और कम समय में समराइज किया है. ये अपने आप में बहुत बड़ी बात है हमारे स्कॉलर्स के लिए, जो रिसर्च में अपने आप को बनाए रखे हैं और नया टॉपिक भी ढूंढ रहे हैं. और आज की डेट में नॉट इवन पीजी, पीएचडी के लिए भी यही अप्रोच अडॉप्ट किए जा रहे हैं, जिसमें कि सारे के सारे जो हैं, चाहे सिंथेटिक हो या फिर नेचुरल सोर्स ऑफ ड्रग है, उसमें से भी बायो एक्टिव कंपाउंड के स्क्रीनिंग को लेते हुए इस लेवल तक काम करने का पैटर्न स्टार्ट हो गया है, जैसा कि आपने बताया डिफरेंट अप्रोचेस को लेते हुए. सो मैं इस इंफॉर्मेटिव सेशन के लिए आपको अपने फार्मा लोग के सभी दर्शकों की तरफ से बहुत सारा धन्यवाद देना चाहूँगा, और उम्मीद करूँगा कि आने वाले समय में भी आप ऐसे ही बेहतर जानकारी के साथ हमारे बीच में आएँ और आते रहें। सो आपका बहुत-बहुत धन्यवाद सर। इस कार्यक्रम के को पूरा करने से पहले मैं एक विशेष अनाउंसमेंट करना चाहूँगा अपने सभी दर्शकों के साथ। आज शाम 7:30 बजे से 9 बजे तक एक खास कार्यक्रम फार्मेसी नेशनल फार्मेसी वीक के समाप्ति पे हम कर पा रहे हैं। दैट इज कॉल्ड रूबरू. On this occasion, our honorable panelist, like Professor K.R. Mahadik sir, Professor R.K. Khar sir, Dr. Anil Sharma sir, Vice President, MV Pharmaceutical Limited, and Professor G.D. Gupta sir, Director, ISF College of Pharmacy, Moga, Punjab. Uh, these four are present with us to enlightening with the information query during the live session of this particular. For all the student, teachers, and professionals of the pharmacy profession, definitely join this evening to get better guidance regarding education and career, and of course employment of that particular. Today, come live on Pharmalok YouTube channel and ask your question to our respected panelist, or send to your questions through mail or SMS or WhatsApp Messenger with us. Our email ID is pharmalok20@gmail.com, and WhatsApp number is 955 5778814. Again, I repeat, 95557788814. So, all the viewers and our respected professionals, please join together at the evening 7:30 on the live session of Pharmalok, that is Pharma Rubaru, with our honorable panelists. So, thank you again, uh, Dr. Saurav Sharma sir, to joining with us with the very informative information with our audience. So, thank you, sir.
Thank you. Thank you, Santosh sir. Thank you. Thank you, Amit sir. Thank you, your team Pharmalog. Thank you.